Welcome to this video. We will be covering serology. We have slide agglutination. I'll give you some information on serological testing, ELISA, and Western blotting. So first up, we have slide agglutination. We have in this particular testing a series of slides. Here I just have one just to keep it simple, but know that there is a series of slides. Now on these slides we'll have a drop of saline, we will also have our unknown bacterium, and then we will also have our known. Let's keep it, let's do that color. Then we have our known antiserum. Now, if we see with this combination clumping, so if we see clumping, or agglutination, then here we have a positive test. Here's some info on serological testing. Now, it's not only there or used to differentiate between microbes such as strep and staph, but between like strep and strep, between the specific species within that specific species. So we have serotypes, serovars, and biovars. Now, these are the definition here is strains with different antigens. Now, antigens is a substance that cause antibody formation. So an example here, we have various serotypes of streptococci. Now, within the cell wall, for example, there are different types of antigens. Each specific antigen can then cause a formation of specific antigens or antibodies to the antigens. So we have our antigen 1, within streptococci, antigen number 2 within streptococci. Now, when it's present in the host will then produce an antibody. But not just any antibody, a specific antibody for antigen 1. The same thing will happen here. A specific antibody will be produced for antigen 2. ELISA, enzyme-linked immunosorbent assay. So we have our wells, a series of wells, and in these wells, we have our antibodies. So these are our antibodies. And these are known antibodies. And known meaning that they know the bacteria that cause the response to within the host to create antibodies. So we know the bacterium. So now we have our antibodies that's fixed at the bottom of the well, on the well, in the well. And then we have our unknown. So our unknown bacterium will then be placed inside of the well. Now, we will see, hopefully, agglutination. That is clumping. So we'll have our unknown bacterium and they will clump together. They will attach to the antibodies. If this occurs, of course, we'll have a positive test because we know then that this bacterium is the bacterium that allowed the host to produce specific antibodies for it. Now, an example here is the AIDS test. Western blotting. It is another test that helps to identify antibodies in a patient's serum. An example here is the HIV infection. They are confirmed, this infection is confirmed using Western blotting. So I'll cover these steps. So step one. Step one, we are using electrophoresis. Now within electrophoresis, this process, we have our lice bacteria that is placed within the instrument or in the gel. And then there is an electric current that flows through the gel. Now, based on the charge and the size, will determine where we will see the pieces of the lice bacteria, or specifically proteins, or even more specifically, the antigens, where they will land within the gel. So number two. So the bands are then transferred to nitrocellulose filter. So there's a transfer. Of bands to nitrocellulose filter. And you see that here. Number three, the filter is then washed. So the filter is washed. Now, what is the filter washed with? Great question. So remember, we know that there is antigens. There are antigens on the filter because it was transferred from the gel to the filter. So remember that we know that there are bacteria there. There are antigens there. So then the first washing would be the patient's serum. After that is another washing of the anti-human antibodies. Now, these antibodies are then tagged with enzymes. So now we have the patient's antibodies. We have the anti-human antibodies with the enzyme that is tagged on it. We also have the antigen, specific antigen. Well, this all will give us visibility once the substrate is added. Need more help? Click the link in my description box to get started with your microbiology study module.